thank you, Eric, for that really wonderful introduction. So it's exciting to be back here in Wisconsin, where I'd say I really cut my teeth on research. And so you've heard a little bit about the bad things that small mammals do carrying Lyme disease, but hopefully today I can talk about potentially some of the good things they do. So small mammals, which you're probably all familiar with, mice, voles, and chipmunks, are often really abundant consumers in forested ecosystems. And I'm really interested in how their resource use, specifically the consumption of mycorrhizal fungi, coupled with their habitat affinities for living in different locations, how those interactions influence ecosystem forest health and function. So in actually worldwide, about 90% or more of all plant species associate with either ectomycorrhizal fungi or arbuscular mycorrhizal fungi. And most all trees, in this region especially, pretty much everything associates with either one of these two species. And hereafter I'm going to just refer to them as ECM or AM fungi, because they get to be kind of a mouthful. Now these fungi colonize the roots of trees and they extend their hyphae out. And they're really good at capturing nutrients like nitrogen and phosphorus, and they also allow trees to access water. Now this is a mutualistic interaction between trees and mycorrhizal fungi. So in exchange in getting these, all these nutrients, the trees are providing the fungi with sugars in the form of carbohydrates. And so many trees cannot grow or survive without these fungi, and so they're really critical. Additionally, maintaining a diverse fungal community is really important for maintaining forest health. So different mycorrhizal fungi are active under different conditions. For example, some might be more active under drought conditions, where others are more active under really wet conditions. They occur at different levels in the soil. And so the more different types of mycorrhizal fungi you have in the forest, the more resilient forests are to disturbances or changes. And as our climate changes, this becomes more important. And so these fungi are acting much like a buffer. Now, maintaining diverse mycorrhizal communities, especially after we have some type of disturbance where a tree dies, the mycorrhizal fungi die, we need some way for these fungi to get around. So there's two main types of fruiting bodies that these fungi produce, called sporocarps. And you're probably all familiar with above ground fruiting bodies, commonly referred to as mushrooms. And we also have truffles, and these are simply below ground fruiting bodies. Now, mushrooms themselves tend to be wind dispersed, but actually they're not very good at dispersing. So especially in forests where we have relatively low wind turbulence, about most of those spores, 95%, only go 1 meter from that spore cap. So mushrooms, although they may be wind dispersed, are not getting very far. And truffles occur below ground and have completely lost the ability to disperse spores on their own. And this is where these small mammals come into play. They find the mushrooms and truffles, they eat them, they swallow these spores, and then they disperse the spores to new areas with their poop. And so it's kind of like a pollination system, but maybe a little more gross because there's a lot more poop involved. And so this is also a mutualistic interaction. So these small mammals are also getting a major food source from eating these fungi. In some cases, it can make up the majority of their diet. So you can imagine that depending on the type and the amount of fungi that these small mammals are eating and the locations that they're depositing them in can have really important implications for the growth and establishment of forests. So especially after we have a timber harvest where these trees are killed along with their mycorrhizal fungi, you need some mechanism for these spores to get back in. And small mammals may play a really important role in facilitating the dispersal of these fungi. And so much of my work has, for my dissertation, has really begun to untangle all these complex interactions that are taking place. And today I just want to focus on kind of 4 vignettes, if you will, on some of these interactions that I've looked at. So the first will be what factors help drive selection of truffles by small mammals? What are they looking for when they're searching for these truffles? The second one will be how does resource availability shape fungivority? So how does masting, or the production of seeds by trees, which Eric has set up a really nice introduction for, how does that influence what they're eating, how much fungi, along with competition between small mammal species? I'm also going to talk a little bit about which small mammal species are the best dispersers of fungi relative to each other. And then finally, I want to end on some of the postdoctoral work that I'm doing now that looks at the role that small mammals may play in dispersing fungi and how that contributes ultimately to fungal diversity and tree establishment in forests. So before I kind of get into some of these questions, I want to give you a background on how I go about collecting these data and where they come from. So much of my dissertation work focused at Bartlett Experimental Forest, which is in the White Mountains of north central New Hampshire. And in that area, we have established a number of sampling grids. They're about 1 hectare in size. And this is, I should mention, a really beautiful area to work in. It's a little more mountainous than around here, so that's a little bit to get used to. And so these grids that we're working with are stratified across the dominant forest types, which we have 4 in hardwood-dominated forests. These are maple, mostly maple and beech. We also have 4 in softwood forests. These are hemlock and spruce-dominated forests. And then finally, we have 4 that are in mixed forests that have hardwoods and softwoods. So before I started working in New England, we really didn't have a very good idea of the diversity and abundance of truffles. And so when I began, I wanted to get a better idea of what's out there. And so we set out to survey across these forest types. In total, we sampled 768 4-meter square plots across those forests for a total of about 3,000 square meters. So this is almost half a football size area that we sampled. So it was maybe a little bit more than we had bargained for. So I really appreciate the help of these undergrads that had no idea what they were signing up for. So to sample for these truffles, we raked to a depth of 10 centimeters using these garden rakes and extracted all the truffles that we found within the first 10 centimeters or until we reached mineral soil. So we really had no idea what to expect when we started this, and we were quite surprised when in this one summer of sampling we recovered over 6,000 truffles of 14 species, comprising 7 different genera. And of those 14 species, it turned out that 5 of them were new to science. They'd never been described before. And, a couple years ago, I was able to describe a number of these new Elaphomyces species. And so Bartlett Experimental Forest is one of the most well-studied forests on the East Coast, established in 1933. And so to find 5 new species that researchers have literally been walking on for decades was pretty exciting. We also found extremely high truffle biomass, especially in this mixed and softwood forest, about 10 kilograms per hectare compared to only about 3 kilograms per hectare in hardwood forest. Now to put this in perspective, in other parts of the world where truffle diversity has been looked at a lot, in Australia and the Pacific Northwest of the US, you usually find about 3 kilograms per hectare. So we're finding about 10 times more biomass in these forests than in other parts of the world. So we have a lot of truffles out on the East Coast. We also found that the most common species, the Elaphomyces species, were associated with eastern hemlock. And this is a tree species that's in severe regional decline on the East Coast due to the introduced hemlock woolly adelphid. So loss of eastern hemlock may have some pretty big implications for loss of truffle diversity and biomass and a potential food source for many small mammals. All right, so on these same grids, we also sampled small mammals using mark recapture techniques. We set Sherman live traps out there. We trapped in 2013, 14, and 15 during the summer months. Each animal was marked with a unique ear tag, a little tag that went in the ear so we knew when we caught it, or a passive integrated transponder or pit tag, which was inserted underneath the skin. We were then able to scan them kind of at the grocery store to see if we had caught them. So these are really good for animals that don't have much ear that's not going to hold a tag. We also collected hair and scat samples to look at diet, and I'll talk a little bit more about this in a second. So for the purpose of this, I'm going to focus on rodent species that made up about 98% of all our captures. And these were 5 different species. It looks like the names are cut off here, but that's all right. You just need to recognize their pictures. So we have the southern redback bull. And I should mention that these guys really live in different habitats. So we have the southern redback bull living in those conifer-dominated stands. We have woodland jumping mice that are more in the hardwood forest. And then finally we have chipmunks, deer mice, and white-footed mice. And these guys are in softwood forest, they're in hardwood forest, they're in mixed forest. They're pretty much everywhere. You probably have them in your backyard, or at times maybe some of these guys in your house even. So these guys are kind of more generalists in their habitat affinities. And so we're really interested in how much fungi are these different species eating. And to get at this, we use stable carbon and nitrogen isotopes. And essentially, these are acting as biological tracers. Essentially, you are what you eat. And so to give you an example of this, I have carbon-13 here on the x-axis and N15 on the y-axis. And you'll notice that the different food species at Bartlett that are available to these small mammals really fall out quite differently in isospace. So we have beech nuts down here, relatively high in C13 and low in N15, and red maple. We have our 2 main types of fungi, arbuscular mycorrhizal and ectomycorrhizal sporocarps. These are primarily truffles. We have arthropods, or insects, over here, and berries over here. So they really fall out nicely. And we can run hair samples for isotopes and compare them to the food source isotopic signatures and use some fancy Bayesian mixing models and actually get the percent contribution of fungi that comprises their diets. And what we found is pretty interesting. So the redback bull, a species that lives in those softwood forests, is primarily eating fungi. About 60% or more is coming from fungi alone. The other species also consume fungi, on average somewhere around 15 to 20% of their diet. So they are eating fungi, but probably a third less than the redback bull. So this gives us a really good idea of how much fungi they're consuming overall, but what types of fungi are they actually eating? And so for this, we used those scat samples that we collected. In total, we analyzed over 1,200 scat samples. These are freeze-dried, they're ground to a fine powder. We push them through a 125 micron screen, so this is about twice the width of a human hair, so it's quite small. That allowed us to collect any spore isolate that was there. And then we used the microscope, looked at 25 fields of view, and counted all the spores that we found within there. And so you can see that these spores, this is Hydnotria, this is Octaviania, these are 2 types of truffles. They tend to be relatively distinct, and we're actually able to identify a lot of these spores, the species or the genus level. So after probably at least 2 years on the microscope and a lot of help from undergrads, I finally had some results. And we found in total about 34 different types of mycorrhizal taxa in their scat. And about 75% of those were from truffles. And so they're primarily eating truffles, although they are eating a few types of mushrooms. And I think if we did genetic techniques, a lot of these we were only able to get to the genus level. If we were using genetics, I wouldn't be surprised if this number would be 2 or 3 times higher. So these small mammals really are carrying a lot of different types of fungi. On average, individuals were carrying somewhere between 3 and 5 types of fungi in one of their scat samples. And redback bulls and eastern chipmunks carried the most. So on average, an individual had somewhere between 4 or 5 different types of truffle taxa in their scat, and sometimes as many as 11 or 12. The other mice species tended to have a few less, but they were still on average carrying somewhere around 2 or 3 different types of fungi. All right, so we know that there's a lot of fungi out there, and we know that these small mammals are eating them, especially the truffles, but what are they actually looking for? So truffles, as I mentioned, are the below-ground fruiting bodies of mycorrhizal fungi. And they've actually evolved from mushrooms, from having this above-ground fruiting habit to then fruiting below ground. So this has some advantages. So fruiting below ground, you're protected from droughts and freezing conditions. But now you're at a disadvantage for dispersal. So whereas mushrooms can potentially be dispersed by the wind, truffles have completely lost their ability to disperse their spores on their own. They're sequestrate, meaning all the spores are totally encased within this outside layer. And so they require another mechanism for spore dispersal. And they've closely evolved with small mammals for this. So when these truffles reach maturity, they start releasing chemical signals in the form of volatile organic compounds, or VOCs. A lot of them have kind of a stinky or garlic type aroma. And this is actually why many truffles, you've probably heard them shaved over pasta or other foods. This is actually one of for some species in Europe are the most expensive foods in the world because of these odors that they're releasing. But in nature, they're used to communicate ripeness. Small mammals then hone in on those cues. They excavate the truffles, consume them, and disperse their spores. But despite this really close evolution between truffles and small mammals, we really don't have a good understanding of what small mammals are actually looking for. So it could be those VOC signals, those odors that they're picking up on. Those obviously have a role. Fruiting depth might also play a role. So if you're really close to the soil surface, you're likely easier to get than if you're really deep down in the soil. And rewards might also play a role in terms of how much nutrients are available. Although truffles tend to be pretty nutrient poor compared to other food sources like seeds or even some mushrooms. And so we're really interested in using Elaphomyces as kind of a study species or a study group for this. And Elaphomyces is a really widespread truffle genus. They occur on every continent except Antarctica. There's currently somewhere around 50 to 75 described species, and they're quite diverse. And they're characterized, they fruit below ground, and they're characterized by this really thick peridium here encasing this gleba of powdery spores. So small mammals are really after this outside layer. They consume this peridial layer and also ingest some of these black spores as they're doing that and then disperse them. And so at Bartlett, we have 4 common Elaphomyces species that co-occur in mixed and softwood forests. And these are Elaphomyces americanus, E. baroculosus, E. macrosporus, and E. bartletti. And so for these 4 truffle species, we measured a number of characteristics of those spore carps. And the first was the depth at which they fruit below the soil surface. So we actually use the isotopic approach using N15 of soil and truffles to calculate how deep they're fruiting below ground, because when you dig them up, they kind of pop out of the soil, so it can be hard to determine where they're actually fruiting. So we use the isotopic approach to look at that. We measured abundance of these different species using the surveys that I talked about earlier. We measured the volatile organic compounds using mass spectrometry for each of these species. We also looked at the size in terms of how much mass are these small mammals getting by consuming the different species. And then finally we looked at nutritional content, focusing on protein, which is something small mammals really look for when they're gathering food. And so what we found is these species really fruit at quite different depths. So we have Americanus that's relatively close to the surface, Baroculosus that's fruiting about 3 centimeters down. And then these 2 species, Macrosporus and bartletti, fruiting relatively deep in the soil, somewhere between 6 and 8 centimeters down. And so this is about twice the length of most of these small mammal species. So quite a bit of effort to go and dig down to get them. We also found pretty big differences in their abundance. So Baroculosus, quite abundance across these forest types, about on average somewhere around 25,000 hectares or truffles per hectare. So there's a lot of them out there. For example, in some areas we would dig up 4 square meters and find 300 truffles in an area this big. So these guys are quite abundant. The other species, on average, somewhere between 300 and 3,000 truffles per hectare, so not quite as common. So you can imagine if you're a small mammal looking for a meal, you might be selecting for Americanus that's relatively close to the surface, or tuberculosis that's quite abundant. Now, before I get into what these small mammals are actually selecting for, I want to orient you to this. So this is a selection index, and -1 indicates avoidance, that they're selecting against a given truffle species. 0 indicates that you're eating a truffle relative to its availability, and 1 indicates really strong selection. These different shapes here represent the different small mammal species. And what you'll notice is that for Americanus, that really shallow fruiting species, it's really either selected against or just consumed relative to its availability. If we look at tuberculosis, that really abundant one, we find that most species are really selecting against it. They're avoiding it. And with the exception of the redback bull that's eating it relative to its availability. And if we look at the last two species, those deep fruiting species, they're actually selecting for them quite a lot. They prefer these species. And so that's a little surprising given how much they would have to work to dig them up. And so maybe it's the volatiles that really help with this. So we measured the volatile compounds, and this is an NMDS ordination. And all you really need to know here is that these different shapes represent different truffles. The closer they are together in space, the more similar their volatile composition is, and the farther they are apart, the more different they are. And what you'll notice is that Americanus and tuberculosis tend to be relatively similar in the volatiles that they're releasing. Bartletti and macrospores tend to be relatively distinct. And if we overlay the volatile compounds, and there's quite a lot of them, these vectors indicate a given volatile, and their strength is indicated by the length of this arrow. So you can see that we have two kind of really distinct volatile communities, one for Bartletti and one for macrospores. And I could try to name off all these different compounds, but I probably couldn't pronounce them. So I thought it'd be easier to show you what these might smell like. So this is an odor word cloud, and I'm taking the volatiles and multiplying them times what we perceived as a human, what they would smell like. So macrosporus really has this sweet component with maybe a hint of truffle and floral fruitiness. And I would actually describe them as kind of a citrus aroma. Whoop. Bartletti tends to also have this sweet component, but also has a truffle, fruity, alcohol, floral aroma as well. And these tend to be quite stinky. And so these deeper fruiting species seem to have more volatiles and a much more pungent aroma. All right, but what about that reward that the small mammals might be getting at? So we looked at the mass and protein content. And so it turns out that these Elaphomyces species are all relatively similar in size. They're about 1 gram, probably about the size of a quarter or so. However, we did see some big differences in their protein content. So this is the amount of digestible protein, and you'll notice that macrosporus and tuberculosis tend to have nearly twice as much digestible protein than the other two species. And so this really doesn't match up too well with what we see. So most of the species are selecting for macrosporus, but they're really not selecting for tuberculosis, even though it does have a lot of protein content, with the exception of the southern redback bull that is consuming fungi. So as you recall, this is a fungal specialist, and so if you're making a living off of eating fungi, you better be selecting for at least a few of these that do have a lot of rewards. And so just to summarize this section up, strong VOC emissions seem to allow these small mammals to detect truffles that can fruit quite deep in the ground. And this stronger VOC composition, although they're fruiting deeper in the ground, it may be easier for these small mammals to hone in on them. So although it's more digging, it's less foraging time looking around trying to pick up these other truffles that don't smell as strongly. And protein content, or that reward, is likely probably not too important, with the exception of that fungal specialist that is relying on fungi as a major portion of its diet. So if you're making a living off it, you do need to get a certain amount of protein. All right, so moving on to this next section. So this was actually a part of my dissertation looking at niche breadth and overlap of two Paraniscus species and how that fluctuated with masting events of American beech. But what I really want to focus on today is how fluctuations in this food source, masting, influences the diets of the small mammals, especially how much fungi they're eating. So in New England, beech is a really common tree species, similar to oak here. It has these masting events where it produces large amounts of seeds every few years, and these tend to be punctuated over time. Beech nuts themselves are a really high quality food source. They're really rich in lipids. They have high protein levels. They fall directly underneath the tree, so if you're a small mammal, they're really easy to gather. They store really well, so they're a really great food source. And it just so happened that during my trapping, I hit two events, one where a lot of beech nuts were falling, and then another where there was hardly any beech nuts available. So this is a really nice test of how these diets are changing relative to fluctuations in food sources. And in this same area, we have two common peromyscus species. These are the deer mouse, Peromyscus mammiculatus, and the white-footed mouse, Peleucopus. They're congeners in the same genus, and they're thought to be relatively ecologically similar, occupying the same habitats and eating the same things. And they're generalists, as I mentioned before. They live pretty much everywhere, including your house, and they eat a lot of different things as well. All right, so I talked a little bit about how we can use isotopes to look at diet, and we can also measure the niche breadth, or what they're consuming. So you can imagine that if they're eating one or just a couple different food sources here, they're going to have a relatively small isotopic niche. However, if you're eating a lot of different food sources, you're going to have a relatively large isotopic niche. And so we can actually use isotopes not only to look at what they're eating, but how large and how large their niche is and how they're overlapping with other species. And so I'm going to start out with some results here from when masting was available. So this is a time when beech nuts were highly available. And you'll notice that these dots here, these black ones, are from the white-footed mice. These open circles are the deer mice. And these ellipses you see are their isotopic niche. And what you'll notice is that this isotopic niche, when mast is available, is fairly small, it's constrained, and there's pretty high overlap. During this same time, they're also eating beech as a large portion of their diet, somewhere between around 33 to as much as 75% of their total diet. And the amount of fungi that they eat is relatively similar between the two peromyscus species, and it's relatively low, somewhere around 12% for ECM and 6% for AM fungi. Now, if we compare this to a season in a different year, where beech nuts are now not available, you'll notice that the niche breadth of these two species is now quite a bit larger. So they're eating a lot more different types of things. And you'll also notice that despite having relatively large niches, about 3 to 4 times larger than these, there's relatively low overlap. So now they're partitioning or competing for resources and eating different things. During the same time, you'll notice that the amount of beech nuts is relatively small, on average somewhere around 5%. And the amount of fungi that they're eating now almost doubles. So for ectomycorrhizal fungi, they're still eating about the same as each other, but on average are eating about twice as much. Same for AM fungi, although it looks like the white-footed mice is eating about twice as much AM fungi than the deer mice. So these pulses of resource are strongly influencing what they're eating. In particularly, the seed availability is likely impacting their interspecific interactions, so how they're competing and how they're partitioning food sources. And during this time when beech nuts are not available, fungi likely makes up a really important food source. And then finally, this consumption of fungi and filling that gap in seeds might help these two species coexist when resources are relatively limited. All right, so we know that these small mammals are eating a lot of different fungi, and everyone's doing it to some extent. So who is the best at dispersing fungi? And so I guess we have to stop and ask the question, first of all, what makes an animal a good disperser of fungi? And traditionally, it's been thought that fungal specialists, those that eat fungi as a major portion of their diet, are the best dispersers. And so most of the studies that have been done have focused on these specialists. In Australia, these tend to be the tongs. And in North America, they tend to be flying squirrels, especially northern flying squirrels. However, the abundance and the habitats that these animals are living in is also going to influence their role as dispersers. And so this is a recent study on bittongs in Australia. And this is a network analysis, and essentially the length of this bar indicates their importance. And you'll notice that bittongs are much more important in dispersing all these different types of fungi than the rodents. However, this study didn't incorporate the abundance and where these animals are living. And so with this in mind, we're really interested in understanding better the role of diet, habitat association, and abundance of these rodents in determining their role as dispersers of fungi. And so I talked quite a bit about now what these animals are eating. We have the southern redback vole that tends to be that specialist, eating a lot of fungi. And the rest of the species tend to be more generalist, although they still do consume fungi, at least for part of their diet. They also have pretty big difference in their habitat association. So we have the southern redback vole living in that softwood forest, woodland jumping mice in this hardwood forest, and the generalists living pretty much everywhere. We also see some pretty big population fluctuation. So in the first year that I started, beech nuts were relatively low and populations were low for most of these rodents. In the second year of trapping, we had a masting event. And the population of actually these three species, but especially eastern chipmunks and deer mice, increased greatly, about somewhere between 5 and 7-fold. And then finally, in the last year of sampling, the beech nuts were all consumed and populations dropped again. So over this time, we have some pretty big differences in populations of these different rodent species. So to get an idea of the relative importance of dispersal, we used network analysis, and we developed these incident networks that are based on the frequency of rodent-fungal interactions. So we took the number of rodent species on a given sampling grid, multiplied by the number of interactions that were taking place, and that gave us a relative index of how important these a given rodent was to a given to the dispersal of a given mycorrhizal taxa. And you'll notice that just because you're super abundant, if you're not really interacting or eating fungi a lot, you're not going to be that important. All right, so this is what the networks look like. And just to walk you through, these letters here are all the different types of fungal taxa. These bars across the bottom are the different rodent species. And the relative length of this bar indicates their relative importance. And all these spaghetti strings you see connecting are all the interactions that are happening in this network. And so in this network, the jumping mice and the white-footed mice are really good dispersers. Redback voles and deer mice are moderate importance. And then finally, eastern chipmunks are of very little importance. And then finally, these bars across the top are the number of interactions that are taking place. So these species, these fungal taxa here are relatively interacted with quite a bit, and then these over here are not interacted very often, so they're relatively rare. So this is the network that I just showed you for hardwood forest in the summer of 2013. This is when beech mast was not available. And for the purpose of this, I'm just going to focus on these three species because they show very characteristic trends. So redback voles, pretty uncommon in hardwood forest, yet they're still of moderate importance. Deer mice also of moderate importance. And as we move into the mixed and softwood forest, where redback voles are quite common, they become by far the most important dispersers of these fungi. If we compare this to the summer of 2014, this is when the generalists, the deer mice and the eastern chipmunks increased. Now you can see that their importance increased quite a bit. And even as we move into the mixed and softwood forest, where we have a lot of redback voles, these generalist species are still of equal or still greater importance than the redback vole. And then as we move into 2015, when the populations of these generalists decreased again, we found a trend similar to what we observed in the first year. So now redback voles are still by far the most important dispersers, especially in this mixed and softwood forest where they're quite abundant. I also like to draw your attention to those bars across the top. These are the number of interactions that are taking place. And what you'll notice is that when those generalists became abundant, we saw about 2 to 3 times more interactions taking place. So not only are these generalists important, but they're also increasing the total number of mycorrhizal fungi that are dispersed. All right, so just to summarize this section up, which species are the most important at dispersing mycorrhizal fungi? Well, it varies, both spatially and temporally. So redback voles, as a lot of studies have found, that fungal specialists are consistently important for dispersing mycorrhizal fungi, but only where they're abundant, only in this case in that mixed and softwood forest where truffles are very available. When generalists increased in their abundance, they also became really important for dispersing mycorrhizal fungi. And the timing that they're dispersing mycorrhizal fungi might be really critical. So we have these masting events that often occur in the fall. The seedlings then begin to germinate in the spring. And this is a time when getting colonized by mycorrhizal fungi is critical for the growth and establishment. And so, coincident with the germination of these seedlings, we have increased number of generalists. These guys are then dispersing more fungi at a time when these seedlings really need to get colonized. So not only are these generalist species important for dispersal of mycorrhizal fungi, the timing at which they're doing it might be really critical for establishment of the next generation of seeds or of trees. All right. So I want to end on this section that's kind of hopefully tying everything together. So that was kind of my dissertation work. And now for my postdoctoral work, I'm looking at how small mammal dispersal, how they influence fungal diversity, and how that might influence tree growth and establishment following clear cutting. And so for my postdoctoral work, I've moved a little bit north of Bartlett. So to the second college grant, which is just on the Canadian border. It's a really cool managed system, actually owned by Dartmouth, and it's part of the adaptive silviculture for climate change, in which they're putting in a number of clear cuts and looking at how they can manage toward the future to help select for tree species that'll do better into the future. And so I'm kind of piggybacking onto this study where they have established a really nice gradient of these clear cuts across arbuscular to ectomycorrhizal dominated tree communities. So we have 4 replicates, 4 of these 1 acre clear cuts in arbuscular mycorrhizal dominated trees. These tend to be sugar maple. We have 4 cuts that are in kind of co-dominated ectomycorrhizal and dominated systems. So this tends to be sugar maple, also with some red spruce, beech, and birch. And then finally we have 4 cuts that are primarily in ectomycorrhizal dominated communities. So these tend to be spruce fir forest with a little bit of birch. And we're using kind of a multifaceted approach to get at this question. And the first involves live trapping small mammals to get at how abundant they are, where they're occurring in the landscape relative to intact forests versus the clear cuts, and also the spores that they're dispersing. I'll also be setting up a greenhouse experiment that'll be growing seedlings in soil collected from the clear cuts and inoculating them with small mammal scat. And then finally using field exclosures that restrict small mammal activity to look at mycorrhizal community changes over time where small mammals are excluded. So I do have, these cuts were established in 2017 in forests that are about 80 to 100 years old. They put in 1 acre clear cuts, so these are about 70 meters or so in diameter. There's some light thinning around them and some areas where all the trees are maintained. And so we took advantage of these cuts and set up trapping webs, which extended about 30 meters into the forest and to the center of the clear cut. And we sampled small mammals similar to what we did at Bartlett in June, July, and August, and collected scat from these animals at the first capture within a month. And so I do have some preliminary analyses from last summer's trapping. So in total, we captured about 600 animals and from 12 species. But of those 12 species, only 6 of them were common. The most common species were the deer mouse and the redback vole. Deer mice tended to be most common in the arbuscular mycorrhizal communities, whereas southern redback voles tended to be more in the spruce fir dominated forest. Short-tailed shrews, eastern chipmunks, meadow voles, and jumpy mice tended to be relatively uncommon. Now, when we looked at the distribution of captures of these different species across this gradient, so the bottom's cut off here, but this dotted line indicates the edge of the clear cut. As we go 30 meters this way, this is into the clear cut, and this is 30 meters into the forest. And what you'll notice is that the deer mice, the southern redback vole, and northern short-tailed shrews tended to occur across this gradient. And especially for deer mice, pretty much it didn't matter where we were trapping, whether we were in the forest, at the edge, or in the center of the clear cut, they were everywhere. Southern redback voles tend to be about twice as common in the forest as in the cut, although they do still go in there frequently. Southern redback voles, or northern short-tailed shrews, tend to use this edge around the clear cut. Meadow voles, as their name might imply, are occurring in the meadow, or this area in the clear cut where these seeds from a lot of herbaceous plants are starting to grow. And then finally, eastern chipmunks and woodland jumpy mice, despite the relatively small size of these clear cuts, they're only about 70 meters in diameter, they really weren't going into the cuts. And so these guys actually do eat quite a bit of fungi, but since they're not moving them into the clear cuts, they're likely not as important to dispersal mycorrhizal fungi compared to these two species that are abundant. So I do have some very preliminary results from scat analyses. I haven't officially counted them, but I've been looking through some of the slides, and it does appear that all the species that we looked at are carrying mycorrhizal spores, including the northern short-tailed shrew, which is generally thought to be an insectivore, eating insects and arthropods. I found quite high spore loads, especially of arbuscular mycorrhizal fungi in their scat samples. I found both AM and ECM spores, and spores from both mushrooms and truffles. And finally, both animals captured in the clear cut and in intact forest had spores. So it does look like they're going in the forest, eating truffles, and then bringing them back into the clear cuts and potentially depositing spores there. So this next summer, we're going to be looking at the location at which they're depositing those spores by looking at the factors that are influencing small mammal activity. So we'll be measuring a number of microhabitat variables, things such as coarse woody debris, vegetation cover, basal area or trees that have been left behind in the clear cut. There are some seed trees that were left behind, and leaf litter. And so hopefully we can determine what variables are bringing these small mammals back in and help forest managers manage for these small mammals and help them facilitate the dispersal of mycorrhizal fungi. So as I mentioned, I'm also going to be setting up a greenhouse experiment. I'm actually going to do this on Monday as soon as I get back home, collecting soil. So for this, I'm focusing on two species, the sugar maple, which is an arbuscular mycorrhizal tree, and red spruce, which is an ectomycorrhizal tree. These seedlings will be grown in soil collected from the clear cut under 4 treatments. The first will be sterilized soil, so soil that has all the mycorrhizal fungi killed. The second treatment will be soil collected directly from the clear cut, and this will get at what is the role of any fungi that survive in the soil, how are those influencing the colonization and growth of these seedlings. The third treatment will be sterilized soil plus scat. So this will give us the role of scat and the spores that are in there in colonizing these seedlings. And then finally, the fourth treatment will be more of a real-world example where we have some mycorrhizal fungi from the clear cut that are likely not killed, and then coupling that with the scat, which has the spores in them from the small mammals. And so this will give us an idea of the contribution of both the soil and the scat to colonization of these trees. And so for each of these trees, we'll be looking at the percent root colonization, so how many of them are inoculated with fungi. We'll also be looking at above and below ground biomass. So this is not for my study, but this is a study with sugar maple. Both of these are sterilized soil The ones on the right are sterilized soil plus arbuscular mycorrhizal spores. So you can see that the potential for bringing these spores in for the growth of seedlings could be pretty substantial. And then finally, I'll be using genetic techniques to look at the community composition, so what taxa are actually colonizing these roots. And the last part of this project will be kind of long-term, where we've established a number of exclosures. These are 2 by 2 meters in diameter. They're composed of hardware cloth, and they extend from the center of the clear cuts all the way 30 meters into the forest, focusing on these reserves where the trees are intact. They are paired with a control, so these exclosures exclude all small mammal activity, and they're paired with a control directly next to it where small mammals are free to move about. And so we'll be following these over the next few years, monitoring soil fungal communities and also the spore bank in the exclosures relative to the controls to look at how this dispersal of mycorrhizal fungi by small mammals is influencing the forest or the soil community of mycorrhizal fungi and also the spores that are there. And then finally, as I'm sure a lot of you know that have gardens, small mammals not only eat fungi, they also eat a lot of plants. And so we'll be using these exclosures to look at the effects of herbivores, especially things like meadow voles that are eating plants, and how they're not only influencing the mycorrhizal community, but also the plant community that is regenerating. So with that, I would like to thank all the collaborators and supporters that have helped me over the last couple of years, especially my advisor, dissertation advisor, Dr. Rebecca Rowe, and now I'm currently advised by, again, Rebecca Rowe and Dr. Sarita Frey. All the undergrads and field help that probably had no idea what they were signing up for before I gave them a truffle rake and asked them to dig up half a football field. I also like to thank my funding sources, especially the New Hampshire Agricultural Experiment Station. And then finally, my parents for hosting me this week. So it's always nice to sleep in your own bed again. With that, I'd be happy to answer any questions if you have them.
I'm thinking in terms of practical implications for the forest industry and ecological restoration. How could those industries or ecological restoration apply what you're doing?
Yeah, so I think leaving things on the landscape like coarse woody debris, down logs and things like that may really help. So we've noticed, we haven't analyzed it, but we've noticed that the small mammals really like to move along those, and those are also leaving logs on the landscape can act as kind of nurseries for seedlings. And so we have these areas that are shaded, they have more nutrients with these downward debris and small mammals colonizing the fungi. So that could be one implication. But yeah, I think, so back in the 70s, a lot of foresters were actually poisoning out in the clear cuts to kill small mammals because they're eating seeds. And so I think hopefully we've come a long way. I think we're realizing that things are a lot more interconnected, and things are pests in some respects, but they also do play critical roles in other ways. Yeah.
Well, I'm guessing that if you're trying to grow some trees, you fertilize it with mouse poop.
Yeah, I'm sure
Is that kind of the bottom line?
Yeah, I think that's, so these pellets they have a lot of spores in them. They're also really rich in nitrogen and phosphorus. And so in terms of the fungi, they act as a really good place to start growing out. And so, yeah, you're adding fertilizer at the same time.
So I'll quit being disgusted by it and sweep it up.
Yeah, I mean, I don't think you want it in your kitchen or your garage. In the forest, it's good. Yeah.
Since the spores aren't digested, I was wondering when you were talking about the shrews, the short-tailed shrews, that they are insectivores, and you were surprised to find spores in them. But wouldn't the insects they eat spores, and then they would be inside of them?
Yeah, that's a good point, and so there's been one other study that's looked at shrews, and they also found that. And yeah, I wouldn't be surprised if it's insects that they're eating and then getting them.
Right.
But some of these spore loads were so high that I think they probably are, as they come across the sporocarp, they're kind of munching on
Sure.
it. But yeah, I'm sure there is some secondary dispersal there as well. And to your point about the digestion, actually some of these spores are stimulated to germinate after moving through the gut. So a lot of the truffles need to move through the gut before they start to germinate or sporulate. Yep.
If I heard you correctly, you found more spores and so forth in the areas you were looking than in other areas that other people have been looking.
Oh, and for truffles.
For truffles and so forth. To what extent is that what you found and that they weren't looking as intensely as you were?
Yeah, so I think there's one study in the Pacific Northwest that did find comparable amounts, and they were also under hemlock. And so I think hemlock and the Lapomyces are probably the main reason we're seeing this high biomass. The intensity could be part of it, but when we were raking, it would be pretty much every other plot had truffles in them. So I think in retrospect, I probably could have got away with about half the sample size and got the same results, but hindsight is 100%. But yeah, so I think that, I think being really thorough helped us kind of nail down our estimates of biomass, but I don't think it was the reason why we found that many. Yeah.
You were handling some of the rodents barehanded, I noticed, and with Paramescus occasionally being reservoirs of also Hantavirus. Is that a concern?
Not in the Northeast. So we don't have the form of Hantavirus that makes humans sick. At least
They had
you
that.
might know. I don't think so.
It's New York virus, it's not.
Yeah. So I think if I was in the Southwest, I definitely would be
Yeah.
wearing gloves.
There was a Appalachian trail hiker that did die of Hantavirus. And
Huh.
I don't know how far north it was. It was north of Virginia, I'm sure. But-
Okay.
So I just
Yeah, I guess it
cautionary
hasn't
note.
been a, I know in Illinois it's a concern, and then as you move into the north, the southwest, it's a big concern. But yeah, I guess I haven't seen it as an issue in our region. And I find that I get a much better grip with barehanded.
Are all of them edible?
All of the small
The
mammals?
No.
The truffles are edible, but I wouldn't say they're the delicacies that you would find in Europe. So I have eaten some of them, and they have interesting aromas. So some are garlicky, some are like road tar, I would say. Some are fishy. My wife and I were actually at a wedding, and we did some foraging. We did find some tubers in Vermont. So this is actually full-size spaghetti. And so that gives you an idea of the size of these truffles. And so these are the same genus as the expensive European kind, but they're the size of a pea. So they are edible, but you're not, you're gonna be hard pressed to get a pound of them. Yeah.
Well, you were taking off the top 10 centimeters of soil over half a football field
Yeah.
worth of forest. How did you reconstruct that?
Yeah, so we were pretty careful to put it all back together, and within 2 years you couldn't tell that we had disturbed it. And I have gone back to some of those, and we did find truffles brooding again in the same spot. So yeah, initially it was a bit of a disturbance, but luckily that was spread over quite a few acres of land. So hopefully we didn't take too many truffles. I think the small mammals still made it through the winter. That's a good question. Eric?
Ryan, seems to be a lack of any discussion about flying squirrels as being a specialist on fungi. Can you address that?
Yeah, so I didn't catch any. I caught maybe 16. So they weren't, well, and I didn't have elevated traps, so I probably missed them. But I'm sure they are eating fungi to some extent. We don't, well, in the last couple of years, there hasn't been northern flying squirrels in that part of the state anymore. They're mostly southerns. So I imagine they are eating fungi too and contributing, but yeah, the main reason I didn't include them is because I didn't catch them. Yeah.
One, just an amazing amount of work, so great job. But I love the shrew story, and it kind of got me thinking, there's something that's already about 10 centimeters down. I don't know, do you have moles up there? I know that sternals don't really have the equipment for
Yeah, we have hairy-tailed moles.
hairy-tailed moles.
Yeah, and so, yeah, I don't know what extent these guys are going down and getting them below ground. We do find a lot of truffle digs where they're definitely coming from above. But yeah, I imagine if you're a mole, I don't know of anyone that's looked, I guess, at the stomach or the scat of a mole to see if there's spores there. But I wouldn't be surprised if they were. Yeah.
With climate change affecting birds and forests and so many animals and things, is anybody studying, or is that any part of your study, are there changing patterns for these small mammals?
So I think for the small mammals that I talked about today, their distributions probably won't change. Things would have to get a lot hotter for them to move. There are a couple rare high mountain small mammals that I don't really catch that are in this region that could change. But in terms of these, I don't think they're at risk. But a lot of the fungi are really under drought conditions. A lot of them could die, and so I would expect that changing climate could have some implications for mycorrhizal communities. And there have been some studies at Harvard Forest in Massachusetts where they've actually warmed the soil up. They see really dramatic shifts in fungal communities in those areas. So, yeah.
Since this series of talks in the last couple of months has been about human influence, this is a good time to bring up the idea of feral cats again, which bring back so many rodents, mammals.
Yeah, I think there was a recent study, I forget the estimates, but they're kind of off the charts, the number of small mammals and birds that they're eating. Yeah, and actually, so I can follow up on that, in Australia, it's a huge issue because they have a number of marsupials that pretty much only eat fungi, and they've extirpated a number of these species. And so loss of those are fungal specialists eating a lot of fungi, and so loss of those species could have some pretty big implications for the future of forests.
Well, if we could all thank Ryan. I'm sure if you have further questions, he'll be available to answer them up here afterwards. So,
Could I put a plug in for Saturday's Master Gardener's plant sale? It's